Assalamualaikum warahmatullahi wabarakatuh. My name is Nadhira Binti Ahmad Nordin, and I'm going to present about the cytotoxicity study of slimming coffee by using plant bioassay supervised by Dr. Lyena Watty Zuraine Binti Ahmad. Being obese or overweight can harm one's health due to the excessive fat built up in their body. According to WHO, obese is for someone who have body mass index from 30 to above, while for overweight is from 25 and above. Slimming product is the additional supplement that can be consumed by other people to reduce their weight body. And for example, slimming coffee. The problem statement: the increasing of the production of slimming coffee and their overclaim and excessive advertisement about the nutrition that their product contain and the impact that will consumer receive sometimes can bring harm to the consumer, because the consumer tend to believe that the product is good to them, but sometimes they doesn't realize that it actually bring harm to them. So this study is to investigate the potential risk by consuming slimming coffee. The objective of this study is to investigate the morphological structure of Lyena radiata after being exposed to selected product for 24 and 48 hours, and also to investigate mitotic index and aberration rate of alum cepa after being exposed to 24 and 48 hours. For macroscopic parameter, the Lyena radiata will be allowed to germinate for 24 hours before transferred to the selected product for 24 and 48 hours. For every 24 hours, the morphology of the Lyena radiata will be observed and the data will be collected. The microscopic parameter is when the alum cepa root tip will be cut off and transferred to 80% HCl for 4 minutes before transferred to the chiral fluid with chloroform for another 4 minutes, and after that we put on the slide and stain with the 2% toluene blue for 2 minutes. After that we apply cover slip and by using the needle to squash the root tip until the cell of the root tip spread out. After that, we will be observed by using microscope, and the data will be collected, and all of the data, both microscopic and microscopic, will be analyzed by two-way ANOVA. The result and discussion. If you can see from the graph, if you can see from the graph of the root length for 24 hours, it shows significant difference. And it also shown that when the concentration is decreased, that means the root length will be increased. And this situation also same when it's 48 hours, when you can see that when the concentration decrease, it mean the root length will be increased. This is due to the one of the condition that was stated by West et al. In 2004 that stated it is the condition that named osmotic stress condition that inhibit the meristemic activity that make the plant to respond for minimal root length. For mitotic index, if you can see from 24 hours, it show the different, but it just slightly different. But when you see it, when you look at the 48 hour, you can see that one of the concentration in this graph shows slightly decrease in their mitotic index. This is due to the two things, which is the exposure of time and also the concentration that increase. And it was supported by the previous study by the Sabin et al. In 2020 that said when the concentration is increased, the mitotic index will be decreased, and it indicate the presence of the strong toxin in that product. And Pohanka and the study by Pohanka 2019 also stated that time exposure also play the main role to the decreasing of mitotic index. For aberration rate, you can see the significant difference. In that graph, and if you can take a look, when the aberration rate is increased, is due to the increases of concentration. The increases of aberration rate was indicate the strong toxin that contained in that one product. And it was supported by the study also by Sabit et al. In 2020 that said when the concentration is increased, aberration rate is increased, it indicates the strong trend inhibition of mitotic activity. In conclusion, the roots growth was inhibited due to the time exposure and the increases of concentration. The decreasing of mitotic index due to the increasing of the concentration indicate the strong toxin in that product. The appearance of the chromosomal aberration such as C mitosis, giant cell, laggard chromosome was indicated to show that the product contained the strong cytotoxic ingredient in one of the product. Recommendation: further study need to be conducted to investigate more about the ingredient that has been used in the slimming coffee to prevent further cases regarding slimming product as it can harm one life. This is the reference that I was used for this study. That's all from me. Thank you.